All right, so today we're going to talk about pedigree analysis and analyzing these genetic family tree-like things, diagrams that are called pedigrees. So first off, as I said, it's kind of like a family tree. It gives you your genetic history. And the purpose of it really is to determine whether or not your children have a probability of having a specific disease or not. And we do that by looking back at your past family history of expression of particular diseases. So, the first thing that you have to know about constructing these things is what are the symbols that we use when making them. So the first thing is that females are always circles and males are always squares. Okay, so that should be pretty easy. The next thing you want to know is how do we designate mating relationships and sibling relationships. So if you look here at number 1, a horizontal line connecting a male and a female, as I've kind of drawn in red here, designates a marriage, or I shouldn't say marriage necessarily, but a mating event, or that they have mated before. And these are, down here, the resulting children from that mating. Notice they had a boy and a girl from that mating. Number 2 shows you that as well. The only difference is we have a little bit different way it branches off here. In number 2, it branches off like this, which designates fraternal twins. The only difference genetically between fraternal twins and brother and sister and sister, or brother and brother, is that they shared a prenatal environment, which could cause diseases and things like that. The last type looks very similar to the fraternal twins. The only difference is they connect down at the bottom here. That designation shows identical twins. They are genetically identical to each other. They form from one egg and one sperm fusing, and then during mitosis and differentiation, kind of even before differentiation, but during mitosis, the two cells split into one, or one cell split into two, and they split into two new embryos, and so they're actually completely genetically identical to each other. Let me get rid of this stuff. Okay. The next thing, I'm going to change the color real quick here, is how we color these symbols in to determine or to designate if they are affected, if they're a carrier, or if they're unaffected. So affected and unaffected are actually exactly how you would normally see them. Affected is usually filled in or darkened, whereas unaffected is not. A carrier, if we know this information, which we don't always know this, will be colored in half dark and half light, like so. So what this basically means is these individuals are heterozygous, these individuals are homozygous, possibly, and these individuals are also homozygous. Now that isn't a perfect thing, and we're going to talk about why that is in a little bit. But these individuals, we know, if they're carriers, have to be heterozygous. All right, so this is what a whole pedigree chart actually looks like. You can see here we have, this is a mating, and this is a mating, and then these are the resulting children from that mating, and these are the resulting children from that previous mating we marked down before. And the individuals who are darkened in, here, 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 have the trait that we're looking for in this case. So notice that 1, 2, 1 and 2 are siblings, and 3, 4, and 5 are siblings. All right, so we want to determine whether or not, or what type of disorder these are. Is it a dominant disorder caused by a dominant allele, meaning you only need one copy, or is it a recessive disorder where you need 2 copies of the allele in order to have the disease? So the way you can tell this usually, if the disorder is dominant, one of the parents have to have the disorder and pass it down to their children. Usually if it's a dominant disorder, you will see the disorder in every single generation. That's not always the case, but because you only need one copy of the disease allele to have the disease, it's much more common than if you would have the recessive trait. If the disorder is recessive, neither of the parents have to have it in order to have a child that has the disease. So that's the big difference between the two of those. And we're going to look at examples of each of these in a minute. The next thing you want to determine is this a sex-linked trait or is it an autosomal trait? Now, an autosomal trait won't differentiate based on sex, based on if it's a male or a female. The way you can tell if it's a sex-linked trait, specifically if it's a sex-linked recessive trait, is if most of the males have the disorder, and you're noticing that pattern over and over again, then it's probably an X-linked recessive disorder or trait. The only way daughters can have X-linked disorders is if their father has it and their mother was a carrier. Okay? If the father passes it to all of his sons and to none of his daughters, then it is a Y-linked trait. And it's found on the Y chromosome. And that makes sense because dads always have to give a Y chromosome to their sons, but they never give a Y chromosome to their daughters. So remember, as I said before, if you get those differences, then it's probably X-linked or sex-linked or Y-linked. But if there's a 50-50 ratio between males and females having it, then it's probably an autosomal disorder. There's also these rare situations that you sometimes see on the AP Bio exam as well. One is mitochondrial inheritance, and in mitochondrial inheritance, as you can see here, our mitochondria have DNA. In fact, they have a circular piece of DNA. Because remember, mitochondria, it's hypothesized, evolved from early prokaryotes that were very good at doing cellular respiration, that were engulfed by ancestral eukaryotes, and they formed a symbiotic relationship. So they have their own DNA and their own ribosomes, thus they have their own genes. So if there's a mitochondrial disease that's inherited, all of your mitochondria you get from your mother in the egg. You do not get mitochondria at all from your father. So a mother passes her mitochondrial traits to all of her children, but only her daughters pass it to all of her children, or their children. Okay? Another example would be an X-linked dominant. And in that case, it's really hard to tell the difference between autosomal dominant and X-linked dominant. The one way you can is that females, if their father has the disease, a female has to get it. Because the father has to give an X. And if it has a disease on it and the father has that X, the disease with the X on it, or the disease that is on the X, then he has to give it to one of his daughters. Otherwise, he gives the Y and they would make a son. So a lot of times if the dad has the autosomal dominant disorder and the wife does not have the disorder, then he'll never give it to his sons, but always give it to his daughters. Okay, so this looks at an autosomal dominant with genotypes that are filled in based on what we know. So you can see here both of these individuals up top have the disease, and if you were to do a Punnett square on that, which is how you kind of solve these things, big A, little a, big A, little a here, you have a 50% chance of having a carrier, 25% chance of not having the disease, or excuse me, let me rephrase this because this is autosomal dominant. What you'd have here is an individual that is homozygous dominant for the disease. A lot of times if it's a disease, these individuals will die, and we've mentioned that previously. We have two individuals here that have the disease, and then one individual that is phenotypically normal. So two people that have this autosomal dominant trait have a 1 in 4 chance of having a child that is phenotypically normal. And if you remember when we talked about like achondroplasia as an example of this, two people that have dwarfism can have a child that is of normal height, and this is why. So if we look through here, you can kind of see how that dominant trait appears in every single generation as it's passed through. But remember, one of the parents has to have it in order to pass it to their children. In this case, at the beginning, both parents actually had it. Here's showing you another autosomal dominant one. Now we can assign the different phenotypes or genotypes here. We can try. Let me select that. So big A, little a, big A, little a, big A, little a, big A, little a. Now we don't know. These could be big A, it depends. We know these parents have to be big A, little a though, because they have a child that is, in this case, little a, little a. So all unaffected individuals in this case are going to be little a, little a. Because remember, the infected individuals have one copy at least of the trait. So now we're going to give this individual big A, little a. They have to be big A, little a because they have a daughter here that does not have the disease. So this individual had to have given a little a to the offspring, and this one is big A, little a. And now we figured out all of the genotypes of the individuals in this particular cross. For autosomal recessive, remember, you can have two people that do not have traits or express the disease having children that do, here and here. So we know individuals, if it's an autosomal recessive pattern, we assign them little a, little a. So this is the opposite of what we assigned last time, right? Little a means that you have the disease. And here we see the two parents who are carriers having children that have the disease, even though they do not. So we can fill in every single individual as little a, in each of these darkened in individuals. Apologize for my writing. And then what you see here are, what are the possibilities here? Now this one could be big A, or big A, little a, but we don't know, so I'm going to make a question mark here. The reason we don't know is because if you do this, we don't see, the reason I think they're big A is they had 3 children, and they had no children that actually had the trait. Now the probability would tell us they probably should have at least one, because there's a 50-50 shot if they were big A, little a of doing that, but that doesn't have to be the case. It's just like people who have 4 daughters. Probability would say that's not going to happen, but it sometimes does. So we're going to put a question mark there. For this individual, we have to put big A, little a, okay, because they do have children that have the trait. Notice these children only have 10 and 11 only have little a's to give. So their children have to have the disease. If we look over here, we know this individual has to be a carrier, and this individual has to be a carrier because they have a child that has it, even though both of them do not. Okay, so that's basically how we figure out these different types of inheritance patterns. Same thing with this one. This has to be a carrier because we have a child that has it. If they're big A, then they would not be able to have children that have the trait. Okay, so that is autosomal recessive inheritance. So this is an X-linked recessive inheritance pattern. So what I know about this is whenever you know something is sex-linked, you have to assign X and Y chromosomes to them. So the male is XY, the female is XX. Now he has the trait, and it's a recessive trait. Okay, so we're going to give him X little a. She is probably X big A, but we don't know for sure, but we're going to leave her like that. This male is also X little a, Y, and they showed that this one is a carrier, so she's going to be X big A, X little a. Doesn't show the disease, but she's a carrier. Now, if you do the Punnett square between these two, you'll actually notice that you can get a daughter that has the trait. Now, one of the characteristics of X-linked recessive is males tend to tend to have the trait way more often than females do, and the reason is because this is the only scenario in which females can have it, other than the mother and the father fully having the disease. The father has to have the disease, and the mother has to be a carrier to have a daughter that has it. So this female is X little a, X little a. This male is X little a, Y, and then this female is X big A, X big A. So remember, for X-linked recessive inheritance, what you're looking for are individuals or males mostly that have the disorder. Is it possible for a female to? Yes, but the father would have to have it and the mother would have to at least be a carrier. For X-linked dominant traits, we're going to do the same thing. So she's XX, he's XY. So she has the trait, so she's big A and big A little a, probably. We don't know for sure, but then we know he's little a because he doesn't have the disorder. Remember, we're switching to dominant now, so having the big A means you have the disease. So this person is X big A Y. She's X big A X, probably little a. And she has to be because she got the little a from her father, actually. He's X big A Y. She is X little a X little a. Now what's neat about this type of inheritance pattern, as I said, is if a father has the trait or the disorder, he passes it only to all of his daughters, and they have to get it from him because he has to give them that X with the disease allele on it. Here's showing you Y-linked inheritance. So notice this father passes it to both of his sons only, and the sons pass it to all of their sons only. That's classic Y-linked inheritance. For mitochondrial inheritance, we have a mother passing it down to all 4 of her children, and then the daughters passing it down to all of their children. I don't know why there should be a line drawn here. Okay? All right, so let's try a couple. So what type of inheritance pattern is this? What you should probably do is pause the video to try to figure it out on your own, and then I'm going to explain how you can figure it out. Okay, so first thing that I see here, I notice that parents without the trait can pass it down to children, and their children have it. Automatically that throws out, or that stands out to me as being autosomal recessive. So this is an autosomal recessive inheritance pattern. And we can check our work too. So if you look here, these two both have the disease. If they were little a, little a, little a, little a, they only have little a's to give. Notice all of their children have the disorder. Okay? And if you would mark every single one of them as little a that has the trait, and then you can figure out who's carriers and who's not. Okay? So this one's definitely autosomal recessive. How about this one? So what I notice here, check out the characteristics of the people that have the trait. I know that may be hard to see, but that red, but you can see we have only males that have the disorder. What I automatically think of is X-linked recessive. Whoops. And you could test it out by doing your Punnett squares and your crosses. So this male would be X little a Y. This female is X big A. Probably X big A. But you can see here that this male doesn't have it. This female was a carrier probably, that she got that. She has to be X big A, X little a, and he has to be X little big A, Y. And then this male got this little a and this Y, and thus he has the disorder. Okay? There's that one. All right, so now what I notice here is that we never have two people who don't have the disease having children that do. So automatically I think it's probably dominant. Another thing that I notice is that the trait appears in every generation if the parent has the disease. Another telltale sign that it's a dominant inheritance pattern. So this is likely autosomal dominant. And again, you can put in your different, you can assign different genotypes and test it out, but these are just shortcuts to figuring it out. All right, this one. We have a mother who has the disease. She passes it to all of her children, and only the daughters pass it to all of their children. Okay? Classic mitochondrial inheritance. For this one, what you want to look at, who has the disease? Notice all males have it. It's kind of funky right here. Let me erase this. All males have it. And it follows from father to father to father. This is classic Y-linked inheritance. All right, the last thing, and we've actually seen this phenomenon before, I just wanted to explain it, is this is showing you autosomal recessive inheritance. And I can tell because we have parents here that don't have it and they have kids that do have it. And the same thing is true here. We have parents that don't have it and a child that does. But what you want to look at is what happened in this second pedigree here. Notice we have grandparents who have children, okay? And then these parents have kids, so this group right here and this group right here are cousins. So what you see here in the middle, right here, is an example of inbreeding. And why inbreeding can be dangerous. Because if you have a disease within your family, any recessive trait, it can cause them to accumulate. And notice right here, we have a daughter that has the disease, who if they wouldn't have inbred, especially if this is a rare recessive allele, the likelihood of any of their children actually getting the disease, some could be carriers, but actually getting the disease would be very small. So this is why inbreeding is such a big problem. All right, so that's it with pedigrees. If you have any questions, please feel free to come ask me in class.